Hello viewers, let us today discuss about TCA cycle, or as we know it as Kreb cycle. It was discovered by H. A. Kreb, who was a German-born British biochemist. So let us discuss the steps involved and the process of the Kreb cycle. So first of all, what is citric acid cycle or a TCA cycle? It is also known as Krebs cycle, and it is a major energy producing pathway in living bodies. So the cells that are obtained from breakdown of glucose in the absence of oxygen, as in glycolysis. So basically, the cycle is used for synthesis of fatty acid, amino acid, and glucose. It serves as a cycle for the synthesis of these products. So, where it occurs location, the citric acid cycle occurs in the mitochondrial matrix in eukaryotes. In eukaryotes, it occurs in the mitochondrial matrix, and in prokaryotes, it occurs in the cytosol. Okay. All the enzymes of the TCA cycle are in the mitochondrial matrix except the succinate dehydrogenase, which is in the inner mitochondrial matrix. However, in prokaryotes, we can see the reaction cycle occurs in plasma membrane. So, basically, the whole cycle occurs in mitochondrial matrix, and the cycle, the net result of Krebs cycle is that for each acetyl group entering the cycle as acetyl coenzyme A, two molecules of CO2 are produced at the end. So now let us discuss about the cycle. This is the cycle. You can see it. You can zoom it. Basically, there are approximately 8 steps involved. The overall reaction we will discuss in the next slide. What happens here is the cycle starts from the Krebs cycle starts from the condensation of an oxaloacetate and the acetyl group of acetyl coenzyme A. The oxaloacetate react with acetyl coenzyme A and H2O to yield citrate and coenzyme A. We basically what happened, the cycle starts from here, pyruvate, which and it is the end product of the glycolysis, oxaloacetate. So what happened here, the oxaloacetate is then condensation occurs and oxaloacetate is then transformed to citrate, after which dehydration will occur and it will move on to aconitase. Here it is, aconitase. Okay, water is going out, and again then the dehydration will occur and isocitrate is formed. There are enzymes here, aconitase. Okay, then isocitrate is then converted to alpha ketoglutarate with isocitrate dehydrogenase enzymes. The enzymes help in conversion. The oxalosuccinate, then alpha ketoglutarate. Alpha ketoglutarate is converted to succinyl coenzyme A, then succinate, and then succinyl coenzyme succinate, then fumarate, malate, and then hexylase. So the cycle goes on like this, citric acid cycle. Okay, so basically the energy yield from two pyruvate molecules when they oxidize to 6 CO2 via the pyruvate dehydrogenase complex and the citric acid cycle. So what happened, overall reaction, we can see from the glycolysis pathway, glucose is converted to 2 pyruvate with the help of glycolysis, in which 2 ATP and 2 NADH are formed. Then the pyruvate is converted to acetyl coenzyme, 2 CO2 are liberated and 2 NADH are liberated. After that, acetyl coenzyme it comes and from the Kreb cycle, in which the overall reaction will be 2 CO2 plus 3 NADH plus FADH plus GTP plus H2O molecule. So the steps are involved. There are 8 steps. They're explained in this slide. You can pause up the slide and see it in detail. So what are the yield of the citric acid cycle? Now let us see what are the yield that we get from the citric acid cycle. You can see the simple figure here. The acetyl coenzyme, it is you can see it very simply. It is 6 carbon, CO2 liberated, then 5 carbon, and then 4 carbon, and then this. So each molecule of acetyl coenzyme entering the citric acid cycle is the following. Like it will 2 CO2, 3 NADH, like this is the CO2, okay, and 3 NADH, this one, NADH, 1 FADH2, this, and 1 GTP. Okay, so this is the total yield. Single cycle, this much, 2 cycles, this much. So the net yield, because each NADH will eventually produce 2.5 ATP, NADH will produce 2.5 ATP, and each FADH will produce 1.5 ATP. Okay, through the electron transport chain, the overall ATP yield for one acetyl coenzyme is 10 ATP. 7.5 from NADH, 1.5 from FADH, and 1 from GTP. So this will be the total yield that we can look. So what are the functions of acetyl coenzyme? Functions of all the citric acid cycle in overall. Oxidation of acetyl coenzyme to CO2, then formation of NADH and FADH for entrance into the electron transport chain, which will occur after the TCA cycle. Then synthesis of several important molecules we have seen in the cycle, like the succinyl coenzyme A, oxaloacetate, okay, alpha ketoglutarate, and citrate. And how it forms, these are given in the brackets. It is responsible for major share of energy release and supply during aerobic respiration. So what happens due to many functions of the citric acid cycle is also considered to be central hub of metabolism. We can see it as a central hub of metabolism, because without this pathway the glycolysis will not move forward, and the functions ahead, they will also not take place. This is because most of the absorbed nutrient, the fluid molecules are oxidized ultimately with the Krebs cycle, and its immediates are used for various biosynthetic pathways. The pathways we will discuss later on. They are also uh, formed due to the citric acid cycle. So basically, the overall reaction we have seen. Now let us see the references I've taken from these all. So basically, we'll move again to the slide. This is the chain. Here we can discuss some more details, like the oxaloacetate. Here the cycle starts. The cycle starts from here, oxaloacetate. Okay, and then it moves on here. So how many steps are involved overall? Overall steps, how many steps are involved? This so first step will be what? First step will be the condensation of oxaloacetate and the acetyl group. oxaloacetate react with the acetyl coenzyme A and H2O to yield the citrate and coenzyme A. Then again in step 2, isomerization reaction will occur, and in third, isocitrate is oxidized and decarboxylated to alpha ketoglutarate. In fourth, the second oxidative and decarboxylation reaction will result in the formation of succinyl coenzyme A from alpha ketoglutarate. Okay. So in fifth step, the cleavage of thiester bond in succinyl coenzyme A is coupled with the phosphorylation of an ADP or a GDP. In sixth step, the third oxidation step in the cycle, FADA remove two hydrogen atom from succinate. In seventh step, what will occur? The addition of water to fumarate places a hydroxyl group next to the carbon. In eighth step, the last of four oxidation step in the cycle, the carbon carrying the hydroxyl group is converted to a carbonyl group, regenerating the oxaloacetate needed in the step 1. Okay, so these are the 8 steps in total which we have discussed. So these are the yields we have discussed. The net ATP yield will be 10 ATP, 7.5 from NADH, 1.5 from FADH2 and 1 from GTP. So thank you for today. Thank you.